My name is Ido Nofech-Mozes, and I'm a PhD student in Sagi Abelson and Philip Awadala's labs at the Ontario Institute for Cancer Research. Today, I'll be presenting my research on pan-cancer classification of single cells in the tumor microenvironment. Tumor microenvironments are highly complex tissues containing multiple cell types which can be broken down broadly as malignant cells, immune cells, and stromal cells. Single-cell RNA sequencing allows us to study the different flavors and states of these cells, and this can ultimately advance our understanding of underlying tumor biology and behavior. In order to effectively study tumor microenvironments using single-cell RNA sequencing, a critical first step in data analysis is annotating the cell types that were sequenced. The basic pipeline involves taking your gene expression data for each cell and grouping similar cells together through unsupervised clustering. These cell groups are then annotated either manually or automatically. To allow for effective analysis of single-cell RNA-seq data, many methods have been developed for automatic annotation. For example, multiple approaches involving integrating cells from a query dataset onto a reference atlas have been very successful. Likewise, other machine learning-based classifiers and marker-based classifiers have also been developed. However, none of these existing methods have been developed with the specific setting of cancer in mind. And cancer samples present unique challenges, often linked to patient-specific events. In this recent publication of 1 million cells derived from a large cohort of ovarian cancer patients, you can see that when the cells are colored by patient, the non-malignant cells form clusters containing cells from each patient, while the cancer cells tend to form patient-specific clusters. Thus, annotation methods that involve integration to an atlas do not tend to work well with cancer cells. This issue is further exacerbated when looking at multiple cancer types. And as you can see here in this example, which highlights lung cancer cells from a pan-cancer cell line analysis, tumors usually don't cluster by cancer type and actually are spread out throughout the embedding. Furthermore, annotation of related immune cells can be really challenging. In this data set of CD4 T cells, CD8 T cells, and natural killer cells, there are multiple subpopulations of CD8 T cells, which share markers with both CD4 T cells and NK cells. And these shared markers can be really problematic in marker-based approaches and in manual annotation approaches. And given the variety of challenges associated with annotating cell types in tumor microenvironments, we decided to ask whether we could develop a standardized method for pan-cancer annotation of single cells. To do this, we first curated training data, developed a machine learning model, and validated our model's performance in classifying cell types across heterogeneous tumor microenvironments. We named our reference-based classifier scAtomic, which is capable of classifying the different cell types across the pan-cancer settings. To train scAtomic, we gathered publicly available single-cell RNA-seq data from multiple sources. Firstly, we used cancer cells derived from 175 cancer cell lines representing 19 solid tumor types. We also included immune cells from both tumors and PBMCs that had corresponding protein markers through CiteSeq. Finally, we also included other stromal cells derived from cancer and normal tissues. Given the large complexity associated with a pan-cancer reference, primarily due to all the different cell types, we designed a stepwise hierarchical classification approach that reduces some of this complexity. Broadly, you can think of any cell in the tumor microenvironment as being either a blood cell or not a blood cell. Within non-blood cells, you can have stromal cells or cancer cells, and similarly, within blood cells, you can have different subtypes. And within these different subtypes of blood cells, you can have even more subtypes of cells throughout this entire hierarchy. And using a hierarchy, we wanted to develop classifiers at each one of these parental nodes, which are indicated by these red stars, where each classification task, unrelated cell types are first removed, and then more specific labels are given as you move down. And through this stepwise approach, we can focus on classifications of related cell types after all the noise has been removed from other cells. To train each of these models, we first selected differentially expressed genes between cell types that are found in the respective model. We then filtered out features that were differentially expressed, but still expressed at low levels in other cell types. And finally, we took these stable features and inputted them into a random forest classifier. To apply scAtomic to query datasets, each cell starts at the top of the hierarchy. At this stage, cells are first scored for their broad categories and moved to the next classification node. This happens iteratively until a terminal classification is made, and additionally, we score the classification confidence at each one of these classification tasks. And if at any point the classification is not confident at an intermediate step, SE Atomic actually stops the classification and outputs a more broad label. Finally, since some samples can have additional normal organ-specific cells, such as lung alveolar cells, we also added a module to distinguish these cells from their cancerous counterparts. And to do this, we scored upregulated and downregulated signatures of cancer in each cell, and then we split normal tissue cells from cancer cells using these signatures. We then validated SE Atomic's performance in an external set of 225 primary biopsies, and we calculated F1 scores. As shown here, you can see that SE Atomic does a really good job in classifying each cell type with mean F1 scores over 0.9 in each cell type. And remarkably, it's able to predict the cancer type in all these cancer cells, which is really cool. We then explored how SE Atomic's performance compares to 6 existing classifiers, which represent a broad set of approaches. And in this comparison, we show that only SE Atomic is able to achieve strong performance in classifying cell type in cancer cells, while the other tools were also accurate in non-malignant cells, such as blood cells and stromal cells. Additionally, we wanted to compare SE Atomic's ability to distinguish cancer cells from normal tissue-specific cells, so we compared this module to copy number variation inference. In this example, we applied a CNV inference method called copycat, where it predicted one population as being aneuploid in this example dataset. As you can see, SE Atomic also predicted the same population as cancer and found other non-malignant tissue-specific cells, as shown here in blue. When we compared this across our validation cohort, we achieved really high agreement rates with CNV inference methods, although we did see some disagreements in samples where CNV inference was poor, and this often happens in homogeneous cancer samples with very few normal cells that the CNV inference algorithms rely on. To highlight SE Atomic's use in practice, we compared it to a lung cancer sample that was annotated with our method, and we compared our annotations to the author's original annotations. And as you can see, their annotations are quite broad, and there are multiple clusters of epithelial cells as shown in these red boxes. When we apply SE Atomic to this data, we achieved much higher resolution in terms of cell types. Additionally, SE Atomic identified the correct cancer type and differentiated lung normal tissue cells from the lung cancer cells. Since SE Atomic uses a hierarchical structure where relevant cell types are eliminated in a stepwise approach, this allows us to extend any terminal classification node with relative ease. And to highlight this, we used new training data to extend the breast cancer node to include histological subtypes. In an external validation data set of breast cancer, we also found that we were able to correctly subtype 39 out of 40 tumors using SE Atomic. In one ER-low breast cancer sample, which is defined as having 1 to 10% of cancer cells being hormone positive, SE Atomic found a subpopulation of cancer cells that was ER positive. Interestingly, these ER-positive cells, as shown here in blue, did not have strong inferred copy number variation and would have likely been misclassified as normal cells if one were using the CNV approach rather than SE Atomic. Finally, we applied SE Atomic to metastatic tumors and assessed its ability to predict the origin of tumors. In 90% of tumors, we achieved correct predictions and found that our incorrect annotations were usually in cancer subtypes that were not well represented in our training data set. One example of this was a mixed serous clear cell ovarian carcinoma that was misclassified as an endometrial cancer, which is a related cancer type. In summary, we have developed, benchmarked, and validated SE Atomic, a new method to classify cell types of the pan-cancer TMEs. We showed that SE Atomic predicts cancer type accurately, it provides high-resolution labels, and it can adapt to new data and further classify any subtype of cell. In fact, we have newer extended models of SE Atomic now available on our GitHub page. SE Atomic was recently published in Nature Communications, so I encourage you all to read the paper for more. In terms of our next steps, now that we've designed the new tool for pan-cancer cell type annotation, we want to start to investigate some pan-cancer questions that can be interrogated with single-cell RNA-seq. And one of these that we're particularly interested in is inference of cellular interactions and cell-cell communication and their link to survival. And we think that the added resolution of SC atomic will be really powerful to do this at a pan-cancer scale. And with all that being said, I really like to thank my two supervisors, Dr. Abelson and Dr. Awadallah, and my labs for their really important contributions to this project. And thank you for listening to my talk. I hope to see you at the conference.